this afternoon. Good afternoon, CGIL. This week we are pleased to welcome Professor Dr. Dorret Boomsma and Nikki, a professor and a PhD student at Vrije Universiteit, presenting for us. Professor Boomsma is a twin researcher and a quantitative geneticist at the Vrije Universiteit Amsterdam. She is a member of the Royal Dutch Academy of Sciences, the Royal Holland Society of Sciences and Humanities, and the Academia Europaea. In 2022, she became a doctor honoris causa at Faculty of Medicine, University of Helsinki, Finland. In the 1980s, she founded the Netherlands Twin Register together with Professor Orla Bacchi, which formed the foundation for her research into the etiology of population variation in human complex traits, and led to numerous publications and over 70 PhD dissertations under her supervision. Her work has led to national and international recognition, including the Spinoza Prize, which is the highest scientific award in the Netherlands. She initiated and participates in multiple twin and GWAS consortia. And Nikki Hoovers is a PhD student of Professor Boomsma at Vrije and has a background in biomedical sciences and behavioral genetics. Her PhD focuses on the etiology of twinning, and she studies the relationship between dizygotic twinning and fertility through genetically based methods such as genetic correlations and polygenic scores. Another important aspect of her research is translating genetic findings into biological insights by studying and integrating multiple omics data, including epigenomics, transcriptomics, and metabolomics. Today, they will be presenting what we currently know about human twinning and its relation to fertility. So you are welcome to start whenever you're ready.
You're looking sideways on my screen. Are you looking at me? I am ready.
Yes,
oh, okay. Okay. Thanks so much for this very nice introduction. And I'm especially excited to present to a group of people who study other species than humans. So I will begin today with giving you an update about what we know about the genetics of twinning in humans. So not talking about the twin method as a design, but trying to unravel why some women give birth to multiples and most of us don't. So it's of course very important to realize that there are two types of twins, dizygotic twins and monozygotic twins. The dizygotic twins arise after a double ovulation and are independently fertilized. And it's a trait that shows huge global differences. Dizygotic twinning is very common on the West African coast, with as many as 1 in 30 or 40 birds being a multiple bird, and it's very rare in Asian countries. It's a complex trait. It doesn't show a simple Mendelian inheritance, but it's clearly segregating in families and in pedigrees. In multiple species, candidate genes have been identified, which, however, largely, not completely, but largely failed to replicate in humans. And after some decades of doing segregation analysis, candidate genes, and linkage studies, we finally could turn to genome-wide association studies, and this has given us the first insights into the genes underlying twinning in humans. The story is completely different for monozygotic twinning. There's no clear implication in epidemiological studies that the trait runs in families, except for some very rare pedigrees, like think of a few pedigrees worldwide, where the trait seems to be segregating in families. So this is basically the outline of our talk. I will talk a little bit about the history of research into twinning in humans and what we know about its heritability. Then introduce the genome-wide association studies that we carried out, where we documented the first significant evidence for genes involved in human twinning, and also talk a little bit about our work in monozygotic twinning and what we hope to achieve in future projects. And then I'm very happy I can hand over to Nikki, who will talk about her work on polygenic score prediction or polygenic risk scores, and also update you on the new project, on the new GWAS that she is running currently and hopes to complete it over summer. So the Bible almost for our work is this book that was published in 1970 by Balmer, The Biology of Twinning. And it beautifully documents the global differences in twinning rates that I just mentioned to you, and also lists a number of other risk factors. The risk of giving birth to dizygotic twins is substantially higher with increasing maternal age, and there is a very well documented risk to female relatives, which correspond to a heritability of about 28% for twinning in humans. And this estimate was obtained by us by converting the recurrence rates that were published by Bulmer for some quite old data sets to tetrachoric correlations and then doubling the sister correlation to achieve a heritability of 28%. Then a few years ago, our Australian colleague David Duffy found some fantastic large data sets online and estimated the heritability for twinning in very large multi-generation pedigrees. So, and when I say multi-generation, the largest one spans 19 generations, and in the next slide I have an example of such a pedigree. This is from an African pedigree, and David estimated that the inheritance patterns in these pedigrees was compatible with a heritability of around 20%. Since we could not distinguish in these pedigrees between mono and dizygotic twins, although the prevalence of dizygotic twins will be much larger than that of monozygotic twins, it seems quite consistent with the estimate that we derived from the data in the Bulmer book. And then we have a new project in progress in which we look at the brother-brother, sister-sister, and brother-sister correlations for being the parents of twins in register-wide data. And the first results have come in from Norway. If we focus on parents who are the parents of confirmed dizygotic twins, the correlations between brothers are not different from zero. In opposite sex siblings, they're not different from zero. But in sister-sister pairs, they're about 0.15. So consistent with a heritability of 30%. And this is population-wide in Norway. So the first step for a dizygotic pregnancy must be a double ovulation in the mother. And we know there are such mothers. This is an example of a large Dutch family that moved during the 1950s from the Netherlands to the US. And this mother had a total of 25 children, also illustrating the association between higher fertility parity and twinning, which included 5 sets of twins. Our colleague Nick Martin followed a cohort of women for a period of 12 months, and the women selected into the study were all mothers of multiple pairs of dizygotic twins. And each month they came in for ultrasound, and This summarizes the months. Oh, I'm sorry. In the middle, you see each month, and you see for this particular mother that nearly every cycle she has a multiple ovulation. Can be on both sides, it can be more on one side than on the other side, but she is clearly a super ovulator. If we turn to other species, we know quite a bit about the inheritance of twinning in sheep. The heritability of litter size in sheep seems to be quite consistent with the heritability of dizygotic twinning in humans. But if we look at the heritability of ovulation rate in sheep, then the heritability is quite a bit higher. So if we could consistently measure this in humans, it would probably be easier to get to the genes for twinning than we currently have to do with a phenotype that depends on the mother actually giving birth to twins. Of course, there are other steps involved in having dizygotic twins or having twins. It's not only realizing the pregnancy, but also being able or having the capacity to bring a double pregnancy to term. So if we switch focus for a moment and look at what we know about environment and twinning, Darwin already observed that the amount of food affects sheep who on high mountains never produce twins, but when they are moved to lowland, they frequently have twins. And a Finnish colleague who worked in the Netherlands for a long time looked at the role of nutrition in producing twins in what we could call natural experiments, although the experiments are quite harsh. He looked at periods of nutritional deprivation in Finland, Sweden, and the Netherlands. And I'm going to focus here on the Netherlands, where in 1944, 1945, the western part of the country experienced the Dutch hunger winter. This was after a large train strike in the Netherlands, after which the Germans decided, if you strike, we will not transport food to the Netherlands any longer. But what Alder Ericsson could do, he could compare the western part of the country where there was very severe deprivation, and then you should think of about having 700 calories of food intake per day, to the rest of the country. And there is a drop in twinning rate, but this similar drop is also seen in the other part of the country, the eastern part of the country, where there was no caloric deprivation. So Ericsson concluded that nutrition seems to be of minimal importance for producing twins. Other environmental risk factors that are quite well established are smoking and body composition, an increase in BMI, body mass index, and heavy smoking. Both are risk factors, quite well established risk factors for dizygotic twinning, although we don't quite understand the mechanism, as those two risk factors are also associated with infertility. Very well documented, more twins with increasing maternal age, and as I mentioned before, very large global differences. Of course, I label this environment, but we should recognize that most of these risk factors could also reflect genetic differences. So, the introduction of genome-wide association studies in human genetics. Our colleague, at that time working at the Vrije Universiteit in Amsterdam, was brave enough, Hamdi Mubarak, to initiate a GWAS project for human twinning. And the first paper was published in 2016, documenting two solidly replicated genes. And the second paper, documenting a larger number of genes, came out in 2024. So in the first paper, we found two genes, FSHB, follicle stimulating hormone, which for a long time had been thought to be implicated in human twinning. And this is actually a locus that is associated with a large number of fertility-related traits, including earlier age at first child, having more offspring, also earlier time of menopause, but this is mainly because of depletion of eggs in the mother. And a second finding was a gene that had not yet been implicated in human twinning, SMAD3. And this is a gene that is expressed in the ovaries and regulates the response of the ovaries to FSH. We did a study in which we tried to estimate what the effect was of women carrying two risk alleles at each locus, and given the prevalence in the Dutch population, when both variants are present, a woman has a 29% higher likelihood of having dizygotic twins. And the work in human twinning, including some of the work coming from linkage studies on very rare variants, was beautifully summarized in this paper that was published, I believe, earlier this year by our colleague Grant Montgomery, who started out his career in sheep genetics and then moved to human genetics. Hamdi went on to also look at a GWAS for being a dizygotic twin in UK Biobank and identified again two genes, again FSHB, and a gene that is quite close to the SMAD3 gene that we identified in the earlier GWAS. UK Biobank also was an important source of information for us, because an important discussion in human genetics is to what extent our results might be confounded or inflated in GWAS study by assortative mating in the population. And there's one very solid zero correlation for non-random mating, and that is for being part of a multiple bird. So that was really encouraging. So then the second genome-wide association study. In the first association study, we included mothers of confirmed dizygotic twins after spontaneous conception, which is again what we did in the second meta-analysis. But here we also included individuals who were twins, including the UK Biobank study. And this enabled us to include twin cohorts in which the twins were genotyped, but not their mothers. And of course, in the meta-analysis, we accounted for the different bits of information of being the mother of twins versus being twin offspring. Well, this is the result, both from the SNP association meta-analysis and the gene-based test. But the next slide summarizes these results in a bit nicer way, because here also the significant and also some of the promising findings, I have to admit, have all been annotated. And what is quite remarkable is that in a lot of projects for human traits that are the topic of GWAS studies, the significant results quite often include a large number of findings in gene deserts of in-between genes, whereas we tend to find for human twinning mainly very meaningful results. And Nikki will talk a little bit more about what this might mean for the polygenicity of human twinning when she is presenting. So as this talk is also recorded, I have included some overview papers on the biology and the genetics of human twinning. And the question is, of course, what next? Well, we know there are still more genes to be discovered. We see quite some promising signal below the line of significance, below the traditional line of significance for GWAS. But also we are currently limited to studies in European populations. So multi-ancestry studies are on the wish list, studies that include other omics. And high on the wish list is also to develop methods to run a meta-analysis for twinning that doesn't only include humans, but also other species. So again, our colleague David Duffy in Australia looked at the predictive ability of our European GWAS into other ancestries. So he took the top 26 SNPs that were represented in multi-ancestry databases and correlated a polygenic score based on 26 SNPs with the prevalence of twinning across different populations. And I'm quite impressed by this result because there is a significant or just significant correlation between this PGS and the prevalence of twinning in the population. So we hope to continue this project and collect samples, especially in this part of the world, that's Benin and Nigeria, where twinning is incredibly common. And our colleague from South Korea, Yoon Min Her, is doing a heroic effort in collecting samples from mothers of twins by visiting schools and then taking DNA samples from all the mothers who have opposite sex twins, so we can be quite certain about zygosity. There are also some wonderful studies in non-human species into twinning, and especially the results in cattle are quite exciting as they point to similar loci across species for having dizygotic twins. Sometimes there's a bit of discussion. For example, in cattle, SMAD6 seems to be the important gene. In humans, it seems to be SMAD3. But as I said, high on the wish list is to combine the human and animal studies. And there's one example of such a study, remarkably for a trait, autism in humans and flight time in these two species of cattle. But in a sense, the phenotype is not important here. This paper is really an important one to my mind because it shows that this can be done, run a meta-analysis across species. Nikki has also spent some time in looking into other omics, but this is a field that really needs a lot more resources and studies. We found some exciting results when looking at the transcriptome, whereas for metabolomics, currently, but also I should admit, a limited platform, mainly looking at lipidomics. For metabolomics, not much in the way of significant findings. Now moving to monozygotic twins, and of course I need to begin with maybe the most famous identical multiples that were born in 1934 in Canada, and are the first multiples, the first twins that are known to have survived into adulthood. They were born into a very, very poor Canadian family. The mother already had 5 other children and then also went on to have more children after this pregnancy as well. So the etiology of monozygotic twinning is completely different. It begins with a single ovulation, fertilization, and then somewhere between day 3 and day 12 after fertilization, there is a split. And we basically do not know anything what triggers the split. The prevalence of monozygotic twins is very consistent across time. It's very consistent globally. And yeah, basically it's a big mystery. There is a hypothesis that twins that share one amnion and one chorion are early splitters. And when they have different chorionicity and 2 placentas, that they are late splitters. But although this sounds very reasonable, there's actually very little empirical evidence for this hypothesis. So since there is so little evidence from epidemiological and pedigree studies that monozygotic twinning has a genetic component, we turned to epigenetics. And there's a little bit of a suggestion that epigenetics could be involved. The incidence of monozygotic twins is higher among some congenital disorders that have to do with imprinting disorders. And also we see an increase in monozygotic twinning after IVF, which was quite unexpected. So one example of an imprinting disorders, Beckwith-Wiedemann syndrome, has a prevalence of almost tenfold among patients. There's an almost tenfold increase of monozygotic twins. And for much rarer congenital disorders, this can be even 100-fold increase. So what did we do? We compared genome-wide methylation patterns between mono and dizygotic twins. We believed that dizygotic twins would be the best control group, as they also are exposed to the same prenatal conditions as monozygotic twins. And as a second step, we also compared methylation patterns between twins and singleton individuals, using 2 epigenetic arrays, 2 methylation arrays developed for humans by Illumina. The first array contains about 450,000 probes, the second EPIC about 850,000. We looked in 2 tissues and we replicated in different cohorts of different ages. So this is the paper that was published in 2021 in Nature Communications with Jenny van Dongen as the first author. We started the project in the Netherlands Twin Register, and to our amazement, because we had done a lot of other epigenetic studies for human traits before, we obtained 243 genome-wide significant hits. This is after Bonferroni correction. We then turned to our international colleagues to ask for replication, and we saw a degree of replication that we also do not tend to see in any other study. These are the correlations for results with other studies when compared with the Netherlands Twin Register. So this is 2 cohorts from the UK, one consisting mainly of middle-aged individuals, a cohort from Finland and a cohort from New Zealand. And there is simply hardly any lower correlation when you look at individuals who are much older than the individuals that we did our discovery project in. Also, the findings replicated quite strongly in DNA that was derived from buccal cells rather than from blood. So the meta-analysis gave us 834 differential methylated probes, which is a signature that is almost strong enough to lead to an area under the curve for prediction of 80%. And here you see a comparison of the monozygotic epigenetic signature in dizygotic twins, monozygotic twins, and non-twins, and it's very clear that the signature is unique to monozygotic twins and not to twins in general. So to summarize, we found a signature that persists from childhood to adulthood across different tissues. But what we do not know, of course, is whether this signature represents a cause or a consequence of a monozygotic twinning event. The signature is almost strong enough, certainly for research purposes, to retrospectively ask the question whether a patient with a particular congenital disorder might have started life as a monozygotic twin. This work is done in collaboration with a large number of twin cohorts around the world that have united in a twinning consortium, and you can follow our work through this website, where we also update the publications, for example. And I'm now getting to my last slide before I hand over to Nikki, and that is to tell you that we have a meeting of the International Twin Conference every 1 to 2 years, where there is a session in honor of Balmer, of the Balmer 1970 book, where we always update on the biology of twinning. Thanks.
All right, then I will take over from here. Should now all be seeing my presentation. Yeah, and I also want to start with a quote of Balmer from the 1970s book. He already hypothesized that an explanation of the perseverance of twinning throughout the population could be that the disadvantage of producing twins is counterbalanced by some advantage. So, of course, a twinning pregnancy bears more risk than a singleton pregnancy. And one of the potential advantages could be a protection against infertility. However, as Dorret has already mentioned, there's been some debate about whether dizygotic twinning is actually heightened fertility, because it's also associated with traits such as a higher BMI and higher smoking prevalence and higher maternal age, which are typically traits that we would associate with infertility. And also in our own genetics work, we saw that an inclusion of a very small case group of mothers who conceived their dizygotic twins through MARD, which is medically assisted reproduction, completely diminishes the genetic signal in our GWASs. So on the graph on the left, there are 200 mothers approximately who received IVF, for example, and they completely diminish the GWAS signal that we find. So obviously there's something going on between dizygotic twinning and fertility. And in our second round of GWAS meta-analysis, we of course did figure out that we should remove these mothers and only focus on spontaneous dizygotic twinning, and then all these nice GWAS signals pop up. And from this, I used this summary statistics to further investigate the relation between dizygotic twinning and fertility through the use of polygenetic scores. Polygenetic scores are kind of our breeding values. This is a different term that we use because, for humans, breeding value is a bit weird name, of course. So that's why we use polygenetic scores, which basically summarizes all the small effects of all the alleles that people carry in their genome. And usually if you look at polygenetic scores within a population, it will follow a nice normal distribution where some people tend to have a very low genetic risk and some people tend to have a very high genetic risk. And what is very interesting for dizygotic twinning, when you calculate a polygenetic score, you can choose to include all the alleles or all the SNPs that you investigate in your GWAS, or you can choose to only include SNPs that have a high p-value, for example, or very low, very significant p-value. And usually for most complex traits, the polygenic score predicts the best or works the best if you include all SNPs. But for twinning, we actually see if you have a more stringent threshold. So here we only include the 0.001 fraction of SNPs with the highest or the most significant p-value, we see that we can actually explain the most variance between cases and controls. And we also looked at this with another tool, which also predicts that only about 17 SNPs should explain about 1.2% of the overall SNP heritability. Which is really a large number. And usually in complex traits, we see that a lot of SNPs will have a very small effect, but for twinning, we really see that only a handful of SNPs or loci tend to have a larger effect, meaning that dizygotic twinning is probably not super polygenic. And then when we compare the polygenic scores, we divvied up the sample that we had of the Netherlands Twin Register in polygenetic in deciles. And here you can really clearly see on the left graph that the people in the highest decile, so decile 10, have very high odds of being a mother of a dizygotic twin, a naturally conceived dizygotic twin. Whereas in the other, on the right side, if you are in the lowest decile 1 or 2 of the polygenetic score, you have a higher odds of being a mother who received ART, so artificial reproductive treatments. So very kind of contrasting the spontaneous, the mothers who conceive spontaneous and the mother who require fertility treatments. Well, we looked into one more way into this relation with the polygenetic scores. We did this both in the NTR and another sample from Norway, the MOBA sample, where we calculated the polygenic scores for dizygotic twinning in mothers of naturally conceived dizygotic and monozygotic twins, and also mothers who received fertility treatments. And what was very nice, at the Norwegian cohort, we were also able to include mothers of singletons. And then we compared the mean polygenic score between these groups to see if anything stood out. Well, first off, when comparing the mothers of naturally conceived dizygotic twins to controls, which were either mothers of singletons or fathers, or mothers of naturally conceived monozygotic twins, you can really see that the mean polygenic score is really highest for the mothers of naturally conceived dizygotic twins, which is also as we would expect. And then if we compare the mothers who received fertility treatments with the mothers who spontaneously conceived their dizygotic twins, can really see that those mothers who receive fertility treatment have a lower polygenetic score. And if we divvy up these mothers even more, mothers who received hormonal induction of ovulation, so this is the group of mothers who most likely have very inconsistent or no ovulation at all, they tend to have the lowest polygenetic score for dizygotic twinning, also lower than, for example, the controls that we have in our sample. So this really maybe kind of shows that giving birth to a dizygotic twin spontaneously is kind of the opposite of having no ovulation or very inconsistent ovulation. In the mothers of singletons, we unfortunately did not see this very strong difference, but we did find that a shorter time of pregnancy of getting pregnant, was also associated with a higher polygenetic score for dizygotic twinning in the singleton mothers. Another thing we did with the GWAS summary statistic is look at genetic correlation. And again, I really want to focus your attention on this very specific type of infertility, which is an ovulatory infertility. This had a very strong negative correlation with dizygotic twinning of 0.7. And when we look at the Manhattan plots locally, this negative genetic correlation is even up to minus 0.85. And this is very specifically around this FSHB region that is also implicated in many other fertility traits. But you could also see on the previous slides that with other fertility or infertility traits such as PCOS and endometriosis, we also see some genetic correlations. This is all nicely summarized in a preprint that's now out that you can find online as well. And this is kind of a picture of a review that has just been published late last year, where they looked at the overlap between the genetic signal of fertility traits, and FSHB is really a location that always tends to pop up, or very often tends to pop up in fertility research. This is of course all based on GWASs that are done in humans, but also in cattle we are aware that some people are really investigating the genetic basis of fertility. And also in this very nice paper they mentioned that some of the cattle fertility related genes are also ortholess or also found with traits such as age at menarche or age at menopause in humans as well. So it seems maybe there is some common genetic background between species as well. Some of our collaborators also did a GWAS into twinning rates in cattle as well, and they identified this region on chromosome 11, which is the FSHR gene. So far in our previous GWAS average, this has not popped up yet, but now we have done a third round of GWAS meta-analysis and we've increased our sample to about 1.3 million. We basically covered all the twin cohorts that there are in Europe, including all of Scandinavia, Germany, also some samples from Australia and the USA, Iceland, UK. Yeah, basically all we could find in Europe. And here on chromosome 2, the same region, the FSHR, also pops up now in humans. But also you see overall we tend to have a little bit of increase in signal. And most of the findings, again, have either been implicated in other fertility traits before, or for example, the peak on chromosome 13 is implicated in the GWAS for testosterone levels, which again tends to be biologically, or it seems to be biologically meaningful for traits such as twinning. So yeah, we're really happy with these results. And in this third round, we seem to identify 9 new regions of interest for dizygotic twinning. And I'm now in the process of looking into these regions and redoing the polygenetic score analysis, etc., to see if we can increase your findings even more. And then, of course, the big question is always, can we do a multi-species GWAS, the same as they did in the example that Dorret already showed with the autism spectrum GWAS? We would be very interested if we can do something like that for twinning as well, especially since we already see some regions popping up in both species. So some remaining questions that we have is, can we maybe predict the work that we do for dizygotic twinning and other fertility-related phenotypes to see if we can predict who might need fertility treatments? I'm working on a Mendelian randomization study to see if we can explain the paradox between dizygotic twinning, smoking, and BMI and infertility. A very big question is, how does the polygenic score actually look like or work in other ancestries? But of course, we're already making efforts in that. And the most important question for you today is, how can we perform a multi-species GWAS for twinning, and is there maybe some option for collaboration today? So yeah, that's all for me. I want to thank all the people involved in the genetic consortium, of course, and I included my email if you ever have questions about my work. Thank you.
Thank you very much to you both. We'll open the floor now to questions either in the room or any online questions as well. So does anyone have any questions to start us off? Anyone go?
Yeah, I can start. First of all, thank you very much for the great presentation. It was enlightening in terms of twinning. I have maybe a question to start. I missed it, I think. What was the density of the panels that you are using more recently, right? How did the panels look like, the SNP panel? Which are the density used in humans?
So the panels, you say?
Yeah, the SNP arrays.
Oh,
so
yes.
that is a bit cohort dependent, but everyone then imputes the data to the same reference panel. So Nikki, how many SNPs do you currently include in the meta-analysis?
About 23 million, but that's also because this time we opted for a little bit lower minor allele frequency. So we used less stringate we cutoffs for that as well. Usually in human GWAS it's about 11 million or 9 million, I believe.
Okay, that leads to my next question. Maybe it's less relevant now because you have these very, very dense SNP panels. But you always mention individual SNP effects. So I wonder why you are not looking at a haplotype. Or haplotype blocks that would explain more the variation of the genome.
Yeah, so when we talk about individual SNPs, we of course note that there's always the LD block around it that also have some signal, but we always try to find an independent signal. But we do use information like that in, for example, gene-based testing. So when we look at more, a bit larger blocks at that. But I don't think we do a lot of looking at haplotypes, for example.
No, in a sense, that's what we tended to do when we were doing linkage studies. But those were quite unsuccessful, although in retrospect, we also have to acknowledge that they were very much underpowered.
Yeah.
And of course, the haplotype information is used in the imputation steps. Of course.
Reconstruct. That's why when you do the imputation, you have the haplotype libraries. At least in cattle, if you like to explain more of the genetic variation that you have in the genome with the haplotypes, we gain in that sense. Because the haplotype will have between quotes, more alleles than a single SNP, right? SNP will be 2 alleles. If you have a haplotype of, let's say, 2 SNPs, you will have potentially 4 alleles or 4 different forms of haplotype. I just wonder, but you are in another level of density that we have in cattle.
Yeah.
I have a question about cross-species comparisons. Have you looked at litter-bearing species, like swine, for example? Have you seen any overlap with candidate genes for litter size with some of the genes you've looked at?
Well, I think the SMAD6 tend to pop up in, well, at least in sheep that I'm aware of.
Also in cattle.
Yeah, but I don't think we've ever looked at swine or other mammals.
No, we have taken the list of candidate genes from animal studies that was published in PNAS. When we published the first paper, there was a list of 75 candidate genes based on animal work. And in the first GWAS, we found very little evidence that they were of importance in humans. And by coincidence, Nikki and I just discussed yesterday that we need to repeat this exercise.
It could be that litter-bearing species evolved quite separately from humans. But, you know, in swine, for example, litter size is very polygenic, so it could be quite different.
What is the normal amount of litter size in swine? Is that also 1, or does it tend to be more than 1 usually?
Well, 14.
Some breeds are 20.
Yeah, exactly. And that's also why we usually don't really look at cats and dogs, for example, because they always tend to have larger litter size already, whereas cattle and sheep usually also only have 1, which is comparable to the humans as well.
I kind of have a follow-up question on that. Do you guys look at differences between singletons with multiples at different levels? So do you see a difference between multiples of 2 versus multiples of 3, 4, or 5, or does it tend to be very related?
Yeah, this is also an outstanding question that we have, like how does the PRS look in mothers of triplets, for example? Last time I checked, I remember that we didn't really have the numbers to say anything about it, but I was planning on doing a deep dive on that again. And for other higher multiple numbers, they also tend to be induced with ARTs, for example. So that's always the risk of, yeah, maybe there's another method behind it.
Yeah, so the rule in human populations is quite simple. If the prevalence of twins is 1 in 80, the prevalence of triplets is the square, etc. So yeah, the numbers become very, very low.
Interesting. Do we have any other? Okay, go ahead.
So first of all, congratulations for our talk. It's very nice to see what have been done in other fields of genetics. And I would like to make a question about the EVF influence on the genetic profile of the individuals. Have you had the chance to compare the embryo from IVF with your embryo for no IVF for a natural process? And have you have some insights about which genes, which areas are different methylated between them?
Yeah.
Yeah.
A study published out of Norway 2 or 3 years ago obtained some very clear results for methylation differences between IVF singletons and spontaneous offspring. And we have done the same for twins, comparing twins after IVF to spontaneous twins. And to a large extent, we replicate the results that are obtained in singletons, even in different tissue and different age. But the methylation results for IVF do not overlap with the methylation signature that we find in monozygotic twins. But this is work that still, this is in progress, is being written up and not even submitted for publication yet. But the results seem very clear.
Thank you.
Other questions in the line? Shelby?
Okay. Can you guys hear me okay?
Mm-hmm.
Yeah. Okay. Because both of you mentioned a relationship between FSH and dizygotic twinning, I was wondering if you guys know anything about how the expression of the gene of FSH, like, I'm going to word this really badly, but if you know anything about the expression and if it increases like the production of FSH and if any of that is associated with like superovulation or like the overdevelopment of follicles available to be ovulated, and then you have like a mom that is more likely to have more kids because she's ovulating so much.
Yes, it's more or less exactly as you say. Higher FSH levels correspond to more multiple ovulation, and this is how do I say this? This is not only within populations, but might also be part of the explanation for the differences that we observe between different ancestries. And this has then led to a discussion of what the origin of differences in FSH levels might be. Is it genetic? And there's also, a belief is too negative, a suggestion that it might be related to diet. So in the countries where there is a very high spontaneous dizygotic twinning rate, a lot of yams are eaten, and those also tend to induce higher FSH levels. Actually, Nikki very briefly mentioned clomiphene, which is a treatment to induce ovulation. And I believe that the first clomiphene products were actually derived from yams. So it's, but there also have been papers within the African populations that the differences in diet are not related to the differences in that we are seeing for dizygotic twinning.
Thank you.
Ella?
Yeah, go ahead.
So I have another question. Is for the monozygote twins, have you looked at the contribution of the male gamete, or the father of the embryo? Because it happens after the fecundation, right? Have you looked at this, if there is any contribution? And if so, how do you control for this?
There is no contribution when we look at pedigree studies in epidemiological projects. There is never a male contribution, not in dizygotic twinning and not in monozygotic twinning. So that doesn't exclude the possibility, of course, that for monozygotic twinning there might be a male contribution that is not genetic.
Well, this might be something actually to study in cattle, because you have, of course, only a few males that tend to have a lot of offspring. You can see if some males have more monozygotic twins in their offspring than other males, maybe something like that.
Absolutely right. You're in a much better position to address this question than we are.
Yeah, that's why I asked the question, because I thought about something similar to look at it.
Yeah.
So would you be in a position to look at this?
Yeah.
I have a bit of a follow-up question on that. So there's no genetic influence of the male side on that occurrence. Is there any studies on whether the male influence may impact the survivability of the embryos to term, or survivability of one or maybe the other, absorption, anything along those lines?
Yeah, so the epidemiological studies that do not find any male contribution, they look at pregnancy outcome generally, so 2 live children. How many pregnancies in humans start out as multiple pregnancies, and how often one embryo is lost is almost entirely unknown. There's one large study, not an empirical study, but a simulation study, so to say, more mathematical modeling that estimates that 12% of human pregnancies start out as twin pregnancies, but we really don't have data. So whether a lot of twins are lost, the so-called vanishing twin syndrome, and whether there might be a contribution to the vanishing twin syndrome, I have no idea.
Interesting. All right, I'll make a last call for questions online or in the room. And it seems like everyone is satisfied for now. Thank you again so much for the presentation. It's, of course, really interesting to see genetic studies in different species, and it was a great discussion. I think we have a lot of interest. And we will have our last CGIL seminar here next week. So we hope to see everyone back again, but thank you so much.
Well, thank you very much for the invitation.
Ben,